Good afternoon, everyone. Welcome to this special event. It is a co-sponsored seminar between the Department of Biological Sciences and the Department of Molecular and Cellular Biosciences. I am Dr. Shelley Thomas of the Biology Department, and I'm co-hosting with Dr. Nathaniel Nucci of MCB. We are very pleased to have you here today. This session is being recorded. So as you can see from the poster that is up, we have today's seminar, and then next week on Tuesday, April 12th at 9:30 to 10:15 a.m. we have our final seminar series. Today's is a research seminar, which is pretty traditional as far as presenting research, although we do add in a career aspect to it at the end. But Ms. Kendra Flores will be speaking on as a career seminar, and she'll be she's a genetic counselor. She'll be discussing what her job is like, how to prepare for that job, and her personal career path. Dr. Nucci, do you want to announce about your upcoming seminars?
Yeah, please. We have one more speaker in the MCB seminar series next Friday. Dr. Glenda Trujillo from Bristol Myers Squibb will be giving both a talk about her research history and about her career path and about careers in the biomedical industry, biotech world. And it's same time as this talk in room 126 of Science Hall, and on Zoom. We offer them as a hybrid event. So I send out, you'll be getting the Zoom link early next week, like I send all of them to you. Thanks, Shelley.
Yep. In today's seminar, we are excited to have Dr. Kristen Blaby-Haas from the Biology Department at Brookhaven National Laboratory. Dr. Blaby-Haas has earned her Bachelor of Science degree at the University of Florida in microbiology and cell science with a minor in chemistry. She earned her PhD at the University of Florida in microbiology and cell science. At Brookhaven National Laboratory, she is leading several multidisciplinary collaborative projects that build synergy between the Department of Energy, national laboratories, user facilities, USDA ARS, and several universities to address long-standing challenges in achieving sustainable bioenergy, bioproduction, and biodesign. As laboratory research manager and head PI of the quantitative plant science focus area, she leads a multidisciplinary team that bridges structural biology, biochemistry, genetics, genomics, metabolic modeling, synthetic biology, and functional genomics to discover fundamental principles underlying successful biosystem design. Here's the format of this seminar. So for about 40 minutes, she'll be giving her research talk, and then we'll have about 10 to 15 minutes for a Q&A for your research. And then for about 10 to 15 minutes, she'll be talking about her career journey, and then we'll have a Q&A again after that. So we encourage you to write your questions in the chat. Please send these questions directly to me, Dr. Shelley Thomas, and then Dr. Nucci will be asking them during the Q&A. It is best if you write these during the presentation so that we have time to organize them for the Q&A. And also, if you're doing this for participation in a class, please make sure that you include your instructor's name and follow any other instructions that they have given you. You cannot see everyone else's questions as you post them, but we are receiving them. So now for our speaker, I'm going to stop sharing. And I would like to turn this over to Dr. Kristen Blaby-Haas. If you would please share your screen.
All right, thank you, Shelley. Can you see it? All right, perfect. Thank you for the introduction. And thank you guys for showing up this afternoon. I'm excited to share our work with you. And I wanted to share with you a recent project that we just had accepted for publication. So we're really excited about it. And it's also a story that really kind of showcases how my group does research. So we mine data from genomics, from high throughput experiments, and we come up with hypotheses about protein function. And then we take that hypothesis and we go to the bench and we use classic genetics, biochemistry, structural biology, you name it, anything we can get our hands on to test that hypothesis, and then ultimately, hopefully, lead to new understandings in protein function. And so the story I want to tell you today is about a zinc chaperone that we discovered, which is the first time that anyone has ever known that a zinc chaperone actually exists in biology. So it's pretty exciting. So first I wanted to just share with you, in case you didn't know, I'm at Brookhaven National Lab, which is a Department of Energy lab. It's out here in Long Island, which isn't too far from you guys. We're out here, close to the fork here in Long Island, the star here. And an aerial view, it's pretty easy to tell what Brookhaven looks like. There's two big rings. This big one over here, this is what's called RIC, and this is where physicists basically, collide small particles together and do physicsy things. And then over here is another ring where physicists shoot photons at different materials and do experiments. And then over here, this building here, these long structures here are greenhouses. And this is where I am in the biology department at Brookhaven. As Shelley mentioned, I lead the Quantitative Plant Science Initiative, and this is a team-based project where we have lots of different PIs, both at Brookhaven and also at different universities, as well as Cold Spring Harbor Lab here on Long Island as well. And we have lots of postdocs and scientific staff that we're all working together to study bioenergy crops. And bioenergy crops are pretty amazing, and we like to think are potentially a really great solution for our climate and energy challenges that we're experiencing these days. And what bioenergy crops can do, and this is both plants, land plants, but also algae, is that they can basically harvest energy from the sun and convert that into chemical energy. At the same time, they take CO2 out of the air, make it into O2, which I think we all appreciate, and then also turn that carbon into various organic compounds, including oils, which can actually be extracted and converted into biofuel. Now, since the Green Revolution, which happened back in the 50s and early 60s, there's been amazing progress when it comes to at least our food crops. And I love these two charts, which really show the power of science to move forward in the plant sciences, which is that since that start of the Green Revolution, there's been an increase in the amount of food we can make, but at the same time, there's been a decrease in the amount of land that we need to produce that food. Now, while this has been amazing, there are still concerns that with the increasing global population, that perhaps these amazing achievements aren't necessarily good enough moving forward. Now, and this is talking about food, but what about bioenergy crops, which could potentially compete with this land with food? Well, one solution that a lot of researchers are working towards is to target marginal soils or non-arable land for production of something like a bioenergy crop. And by doing this, you could potentially avoid competition between our food and our bioenergy crops. Now, to turn something like what seems like a barren wasteland into a lush green bioenergy field is going to take some work. And we have to be careful, because when it comes to something like a bioenergy crop to produce biofuels as renewable energy sources, we certainly don't want those to be more environmentally damaging than the sources of energy we have now. And agriculture is not a particularly environmentally sustainable practice right now. This is a fairly recent chart looking at various contributions of agriculture to greenhouse gases, land use, fresh water use, and then this one is, of course, quite troubling, which is the contribution of agriculture to eutrophication, which is basically that process where our food crops require a lot of fertilizers. And those fertilizers, ultimately, a lot of it ends up in our waterways, leading to algal blooms and eutrophication and harm to our water ecosystems. Now, in the Quantitative Plant Science Initiative, we're really fascinated about plants and how plants, the nutrition and the diet of plants. And some years ago, over 100 years ago, Justice von Liebig came up with this principle, and it's called the Liebig's Law of the Minimum. And it says that yield, and yield here means basically the growth and the health of a plant, is proportional to the amount of the most limiting nutrient, whichever nutrient it may be. And so what this means is that here on the bottom left here, plants require lots of different nutrients to be taken up from the soil. And although we hear a lot about nitrogen, certainly nitrogen is a major macronutrient that plants must take up from the soil, there's also a lot of these micronutrients that are just as important. And you can be in a situation, or the plant is in a situation, where it can have plenty of nitrogen, plenty of phosphorus, sulfur, all the macronutrients. But if it's limiting for one of these micronutrients, its yield and its health is very, very poor. And here's an example on the right here of crops that are experiencing iron and zinc deficiencies. And so because these micronutrients, however, are needed in relatively small amounts, they've been kind of ignored, at least compared to the macronutrients in terms of understanding plant nutrition. And so our project is focusing right now on understanding how plants and how bioenergy crops respond to zinc and iron in their environment. And in out years, we plan to expand this to other micronutrients and then include the interactions with the macronutrients. We're doing two main approaches in our project. One is a multidimensional omics-based approach where we're taking two bioenergy crops. Here is poplar, which is a tree, and here is sorghum, which is a grass. We're taking these two very different bioenergy crops and we're growing them under lots of different conditions of high and low zinc, high and low iron. We're then sampling both the leaf and the root, and we're asking lots of questions about how those plants are responding to those conditions, specifically at how their genome, how their transcriptome is responding, which genes are turned on, which genes are turned off. What I wanted to talk to you about today, though, is the work that we're doing at the molecular level. And so I'm really fascinated in understanding, as I said at the beginning, how gene and protein function works, and understanding how you go from the numerous genes and proteins that are in an organism to the phenotype that that organism has, how that organism behaves, how that organism responds to its environment. And like I said, I'm particularly fascinated by these trace metal nutrients. So here's a periodic table of the elements, and biology has evolved to require these nutrients here that are colored. I want to talk today about zinc, which is a metal in the D block, and it's an absolutely essential nutrient for all of biology, as far as we know. We have yet to find any organism that can we have not been able to find any organism that can live without zinc. And the reason for that is that zinc is an essential cofactor for lots of important proteins. And here are just some examples: RNA polymerase, the ribosome, carbonic anhydrase, transcription factors. These are all very important proteins for most every organism. Now, of course, our group is focused on bioenergy crops, but like I said, these nutrients are essential for everything, for everyone, and even for humans. So zinc deficiency, for instance, affects 1/3 of the global population. And zinc deficiency is actually one of the lead causes of hidden hunger, which is together with vitamin A, iron, and folic acid deficiencies. Zinc deficiency symptoms can range from neurological disorders to weak immunity to diarrhea. And zinc deficiency is actually thought to be responsible for 500 to 800,000 deaths per year. So this is a pretty significant nutrient. Now, of course, everyone sitting in this Zoom probably isn't thinking about, hey, did I get enough zinc today? And that's because our bodies are really great at making sure that we get enough zinc in our diet and not too much. And really, it's only when we see genetic disorders or mutations in some of these homeostasis mechanisms is it even apparent that a nutrient can be both essential and then also potentially toxic. Now, how organisms from humans to plants deal with metals. So like I said, if you have too much of these metals, it can be toxic. But if you don't get enough, it is certainly just as detrimental. And there's lots of mechanisms to make sure that we have just the right amount. Now, the first mechanism is transporters. So these are proteins that are in the membranes of our cells. And there are different types of transporters. There's one set that actually pumps metals into the cell when they're needed. And there's another set of transporters that actually pump metals out of the cell when they're not needed. And over the decades, various labs across the world have been able to find and characterize lots of different transporters for different metals. Also, there are cases of what we call austerity, where some organisms and some cell types can actually change their requirements for a metal. And this is called both sparing and salvaging, where metal-dependent proteins, if a nutrient is limiting, if that cofactor is limiting those particular metal proteins, if they're not too essential, they can actually be degraded and that metal can then be reused for a protein that is essential. And then also in our cells and in lots of different organisms, there are also mechanisms to sequester and store metals. We have vacuole-like organelles in our cells that are actually able to basically create stockpiles of these metals that can be released in the cell when those particular nutrients become limiting. And then also, if a particular metal becomes too high of a concentration in a cell, very similar mechanisms can serve to quarantine those metals so they don't cause damage. Now, what I wanted to talk about today are metal chaperones, which these are proteins that in some cases there's evidence that they actually can dock with these transporters that are bringing metals into the cell and hand deliver them like little delivery trucks to their destination, whether that destination is a protein that needs that metal or perhaps a transporter that then pumps that metal to another compartment in the cell. Now, we have lots of different examples of these types of chaperones existing. Probably the best characterized ones to date are the copper chaperones. Like I said, these are proteins that are thought to actually dock with the copper transporter in the membrane and then hand deliver that copper to its next destination, which for instance could be an enzyme here showing copper zinc SOD, which is involved in oxidative stress. There are also cases of, we don't typically call them chaperones, but they do kind of function like a chaperone, which are the cheilatases. And these are proteins that in the case of iron chelatase, it actually takes iron and puts it into this protoporphyrin ring here to create heme. Whereas this protein here called magnesium chelatase does the exact same thing, except for that it puts a magnesium here, and then this molecule becomes chlorophyll. There are also other cases of nickel chaperones. This, for instance, is a protein called urease. It actually needs a whole suite of proteins to deliver and insert nickel into this enzyme for activity. Now, zinc. So, well, does such a protein exist for zinc? And for the longest time, we didn't know. We had never found a zinc chaperone. So therefore, we made the assumption that, well, zinc chaperones do not need to exist. Okay, so enter the stage, this family of proteins. This is a large family of proteins called nucleotide-dependent metal chaperones. And these are proteins that are related to known nickel and iron chaperones. So one of the proteins that's needed for urease maturation is called UreG, which is from this family. All of these proteins have conserved GTPase and metal binding residue. So GTPase is basically an enzyme that uses the hydrolysis of GTP, which is just a small molecule in the cell, to energize structural conformational changes. And often we see that these proteins are expressed under zinc deficiency, which is curious and interesting. And then in various studies, scientists have been able, including myself some time ago, have been able to knock the genes out that encode some of these proteins in various bacteria. And we often see growth defects when you then grow that mutant under low zinc, which is consistent maybe with the role of a zinc chaperone, but certainly not definitive. Okay, so for the longest time, we and others have thought that perhaps some of these proteins are zinc chaperones. But if it's a zinc chaperone, then it must be delivering zinc to something. And that something has been unknown for a long time. So to actually prove that these are zinc chaperones, we need to find what it delivers zinc to. And to do that, I like to mine data. But in order to mine data, you have to have a really good understanding of the phylogenetic relationships among proteins. And I decided to look at eukaryotes and what data we have for these eukaryotic proteins, because in eukaryotes, it's very clear that there's a single family. And except for very recent duplications that are specific to the human genome, almost every eukaryotic genome encodes one single protein from this family. And whenever you see that type of relationship, it strongly suggests that there's a conserved function across those different organisms. And in this case, perhaps a conserved protein that it's interacting with. And once you have an understanding of how a family of proteins is related, you can then look at lots of different data types from high throughput studies, from genomics, to inform on what could be a function for the whole family. And it's kind of like a detective story where you're collecting lots of different pieces of evidence and trying to understand what is the evidence pointing to. And this is a summary of that work. This is an ortholog-based network analysis where each of the nodes, the dark nodes here in red, purple, orange, these represent a protein from this family that I'm interested in. These are the putative zinc chaperones. And all the other circles represent proteins and genes that this protein is interacting with. And the interaction could be physical. So there are experiments where we can actually do high throughput studies to find physical interactions of two proteins interacting together. And then also genetic interactions where if you knock out one gene, you may or may not see a phenotype. And if you knock out a second gene in that background, the phenotype can actually be exacerbated or relieved. And when I did this type of analysis, I was saying, do I find any data that's the same for every organism? And I was able to find one interaction that occurs in humans. It occurs in these two yeasts. This is Saccharomyces cerevisiae, and this is Saccharomyces pombe. All of these homologs all interact with the same protein, which is methionine aminopeptidase type 1. Okay, so what is this methionine aminopeptidase? So this is a protein that it actually binds to the ribosome. And as you know, the ribosome is the protein that makes other proteins in our cell. So the ribosome is a complex of proteins. And as the ribosome is translating mRNA and making a polypeptide, every protein in our cell starts with a methionine. And this is called the initiator methionine. Methionine aminopeptidase, for not every protein, but for most proteins, this methionine aminopeptidase actually cleaves that initiator methionine. And this reaction is thought to be very important for proper N rule protein degradation. So a lot of proteins, their half-life in the cell is affected as whether this internal methionine is present or not. It's also been shown recently that if you get rid of this activity, there's actually translation defects. So there's a lot of pleiotropic phenotypes that can happen if you don't have this protein and this activity. And what was interesting, of course, for our story is that the protein that we find interacting with our putative zinc chaperone requires zinc for activity. And there's actually two, and this is important to know later, is that there's actually two of these proteins in our cells. One, which is the first one, type 1, MAP1, which is zinc dependent, and that's the one I find interacting with our putative zinc chaperone. And then there's a second paralog called MAP2, and that one is actually thought to be manganese dependent. Okay, so by just sitting in front of the computer for a couple days, we could come up with a hypothesis without lifting a pipette or doing any experiments. And the hypothesis is that this protein, the putative zinc chaperone, which we've actually named Zinc1, is a GTP-dependent zinc chaperone. And the GTP-dependent comes from the fact that if you look at the protein sequence of these proteins, they have well-conserved GTPase motifs. We think it's a zinc chaperone that delivers zinc to MAP1, which is the protein we found in human and in different yeast interacts with this protein. And then also in bacteria, we often see these phenotypes associated with zinc deficiency. So perhaps this protein is needed during zinc deficiency. All right, so we need to test it. So my go-to for testing hypotheses is genetics. And this is how this works. So here we're looking at a map of a genotype, a genome. And on this particular genome, there's two genes. One gene encodes the methionine aminopeptidase. One gene encodes our putative zinc chaperone. Our model is that when there's plenty of zinc in the cell, that this enzyme, this MAP1, can probably bind zinc, and the cells are pretty happy. But under zinc limited conditions, when the amount of zinc becomes limiting, maybe this protein is not so great at getting zinc and it needs a helper. And that's what our zinc chaperone, we hypothesize, is doing, that maybe our putative zinc chaperone's really great at getting access to this depleted zinc. And it can actually dock with and bind to this MAP1 protein, and then using GTP hydrolysis actually transfer zinc to MAP1, and the cells are happy. So to test this model, the first thing we want to do is we want to get rid of the putative zinc chaperone. So we can do that with genetics. Here we're going to be using Saccharomyces cerevisiae, where we can do what's called homologous recombination to remove genes. So we can delete the gene that encodes the zinc chaperone. Now this mutant no longer has the zinc chaperone, and our prediction is that under zinc replete conditions, when there's plenty of zinc, the cells probably don't care if the zinc chaperone is there. But under zinc limited conditions, if we get rid of the chaperone, then this protein MAP1 is not getting its zinc. It's an essential enzyme, and then the cells are probably not going to be too happy. Now, if you remember, I did mention that there is this other protein called MAP2, and it has overlap function with MAP1. They both cleave the initiator methionine of nascent polypeptides, but whereas MAP1 uses zinc, MAP2 uses manganese. And so what we would expect is that under zinc limiting conditions, the cells, maybe they're have a bit of a growth defect, but probably not too much, because this MAP2 protein is there to compensate for the loss of MAP1. So what we can do is, if we're right, we can delete this MAP2 protein. Under zinc replete conditions, again, the cell probably doesn't really care if this MAP2 is absent because it's got MAP1 to cleave the initiator methionine off of these proteins. But now under zinc limited conditions, the cells are probably going to be pretty sick because they don't have MAP2 to rely on, and because MAP1 is no longer getting zinc, it can't rely on that protein either. All right, and so like I said, we worked with the unicellular yeast Saccharomyces cerevisiae for a couple of reasons. One is that it has a single MAP1 gene, a single MAP2 gene, and a single Zinc1 gene, the putative zinc chaperone. So it makes the genetics fairly straightforward. And so this is what it looks like. These are wild type cells. So it's got all the copies of the genes we're interested in. This circle here represents a vector. And these are just cells that are plated on a solid agar plate. And then they grow up and form colonies. And what you're seeing here is these colonies growing on different concentrations of zinc. And you can see how even the wild type, when it's got all of these genes, it grows pretty slowly under this very low concentration of zinc. We can then knock out the putative zinc chaperone, and what we see is we don't see a huge difference, but we do see a slightly smaller colonies, and there is a subtle growth defect if we get rid of this putative zinc chaperone that seems to be exacerbated under low zinc. We can then bring the gene back on a plasmid and see that we rescue. So now the cells aren't so sick anymore. So that provides us with confidence that the phenotype we are seeing is due to the deletion of this gene and not something else that we don't know about. We can also then knock out the MAP2 gene, and as we predicted, the cells don't really care if this MAP2 gene is there or not because it has the MAP1. But if we knock out both Zn1, the putative zinc chaperone, and MAP2, we see a really quite severe growth defect in the cells. These mutants just do not grow under low zinc. And again, we can add the zinc chaperone back on a plasmid and see that we revert the phenotype back to wild type. And our conclusion with this experiment is that this Zn1 is needed to support growth during zinc deficiency, and this function becomes essential when there is a MAP2 defect, and this is all consistent with our hypothesis. Now, the genetics provide support for our hypothesis, but they don't really tell us what the protein is doing at a molecular level. And so for this, we did 2 experiments. The first is we looked at in vivo methionine aminopeptidase activity. And how we do this is that the MAP1 protein, if it's loaded with zinc, which it's needed for activity, it'll cleave the initiator methionine of this reporter that we built that's fused to a protein called GST. And you can order an antibody that will react with this reporter if the initiator methionine is present, but it won't react with the reporter if the initiator methionine is cleaved. And then we have another antibody that just reacts with this GST that serves as our loading control. And then we do what's called an immunoblot. And here what we see is that in the wild type cells, we detect no reporter with the initiator methionine present, saying that our MAP1 is fully functioning. We then get rid of our putative zinc chaperone, and we see that under low zinc, we now have a methionine aminopeptidase defect. We get rid of MAP2, cells don't really care, but if we get rid of both of these proteins, we see a pretty dramatic defect in methionine aminopeptidase activity, and this defect does increase under low zinc. And so again,
just a quick note that we're at the 30-minute mark, so you have about 10 minutes.
Perfect. And so again, these experiments are consistent with our hypothesis, and these experiments are actually a little surprising because what we did see is that when you don't have this MAP2 protein to compensate for the loss of MAP1, we see a defect even when there's plenty of zinc. So this started to hint to us that perhaps this zinc chaperone is actually needed kind of regardless of the concentration of zinc in the cell. Now, the last experiment we did is to test the actual hypothesis, which is that this protein can actually literally transfer zinc to MAP1, which is its client protein. And so my postdoc Miriam developed this assay where we take the MAP1 enzyme and we can strip it of zinc. When we strip it of zinc in vitro, it has no activity. We can then take our putative zinc chaperone, we can preload it with zinc. We can then mix the two proteins together. Now, when we mix the two proteins together, we actually don't expect MAP1 to become activated because the chaperone we think is a GTPase and requires GTP for transfer of zinc from the chaperone to MAP1. And so it's only after we add GTP that we would expect to see transfer of zinc and detect MAP1 activity. All right, and here's what these assays look like. So here we take MAP1, we strip it of metal, it has no activity. We can give it free zinc in vitro in the lab in a test tube, and we see some activity of the protein with free zinc. And if we add a chelator at the same time, we actually can decrease the activity because the chelator is binding to that free zinc and keeping it from binding to MAP1. Then what we can do is instead of giving it free zinc, we give it the chaperone that's preloaded with zinc. And we see two interesting things here. The first thing we see is that when we give MAP1 the chaperone bound to zinc instead of free zinc, there's actually an increase in activity, and that activity is no longer inhibited by EDTA. As a control, we can also use a GTP analog, which is called GMP-PNP. And this is an analog that looks kind of like GTP, and it binds to proteins like GTP, but proteins are unable to hydrolyze it. And what we see is that this analog does not lead to MAP1 activity. So this is a nice control to say that this reaction that we're detecting is dependent on the hydrolysis of GTP. Now, of course, biology is very complex. And we are also really interested in understanding why we see the phenotype we see. And we actually have done some proteomics, which is to look at all of the proteins in the cell that we can detect and ask what's happening in the mutant compared to the wild type. And what we found is that actually in these mutants, the zinc response is not turned on. So that seems to be the consequence in this case of not having the zinc chaperone and turning on the MAP1 activity. And so this is exciting. It completely changes how we think about zinc in eukaryotes and opens up a new chapter in understanding zinc homeostasis. And from our various experiments, we now have this model where we think that actually under zinc replete conditions, that a proportion of this MAP1 protein is able to bind zinc in the absence of the chaperone. But for full activity, it does need this chaperone. And then under zinc limited conditions, its dependency on this chaperone becomes absolute. Now, of course, I am leading a bioenergy crop project and we're interested in plants. And so now that we've done all of this work in yeast that was fairly easy to do and fast, and we've taken a protein that was kind of unknown function, certainly in eukaryotes. We figured out a function using yeast as a model. We now are in the process of confirming that yes, that function is also conserved in plants. And it appears to function just like it does in yeast as it does in plants. And with that, I'd like to make my acknowledgements to the Department of Energy, which funds our work. We also have collaborations with various user facilities that, for instance, EMSL here, which did the proteomics for us. The work I presented was done by 2 postdocs from my group, and then also a collaborator, Lee Fong at Cold Spring Harbor, who is working on the plant studies with us. And then also I wanted to mention, if you saw anything today that you thought was interesting and would be a cool research experience, please check out these two websites here in yellow for the SULI program, which Brookhaven National Lab participates in. And so these are undergraduate internships where you get to participate in research projects and you get a stipend, which is pretty awesome. And with that, I'm happy to take any questions.
Okay, thank you so much, Dr. Blaiby Haas. We have a variety of questions from our audience, and I encourage the students, if you have additional questions, please go ahead and continue to submit them. The first couple questions deal with the beginning part of your talk. Do you know if worldwide crop yield and relative land usage worldwide has followed the same trends as we see for the United States?
And so, no, I don't think so. And I think that's certainly one of the issues that we have, is that while the US has done great, that's not true for the places, you know, on our planet where it matters the most, really. And so there are a lot of people who work on farming practices and agricultural practices in various countries to get to that point where, you know, have really high producing crops, but on relatively low land and small inputs.
Okay, thanks. Our next question is, are there synergistic effects on the yield of a plant if there are numerous limiting micro and macronutrients?
Ooh, good question. Probably. That is something we want. So my group is starting to look at, we're planning to look at starting probably in a year or two. Right now, the experiments we're doing is where we're just changing one variable, which is we're just changing one nutrient. And what we're going to be doing is stepping that up and getting more complex and start changing 1, 2, 3 nutrients and see what happens. Certainly, yes, I would. Even though like Liebig's barrel is very simplistic, there are going to be additive effects if you have co-limitation between different nutrients, certainly.
Thanks. So the next question is, are there outside factors that could affect the concentration of zinc in the soil? For example, rainfall or other plants or the degree of slope that can influence soil going from zinc rich to zinc depleted?
Yeah, I didn't talk about it, but when it comes to soils, oftentimes the challenge isn't the absolute concentration of these nutrients in the soil. It's what we call the bioavailability of these nutrients in the soil. And there are certainly different conditions that can affect how hard the plant has to work to take up these nutrients from the soil. Probably our best example is iron. Iron is one of those nutrients that's really not bioavailable under physiological pH in the presence of air. Plants actually expend a lot of energy overcoming the poor bioavailability of iron, for instance. And so that's a challenge also when it comes to fertilization. With a lot of these micronutrients, you can't, like iron again is another example where you can't just take iron and spread it across a field. You actually have to have pretty expensive iron fertilizers that are providing iron in this bioavailable form. So yeah, there are lots of different aspects to soil quality that can certainly affect how easy it is for the plants to take up these nutrients.
So that answers the next question in our list, which was about using high metal nutrient fertilizers. It's like a prescient answer, pretty impressive. So the next question is, do you envision a sort of a golden rice analog for zinc in terms of addressing zinc deficiency? Is that something that's feasible?
Yeah, and I actually didn't present it, but I have another side project where we actually discovered that a protein that's similar to this zinc chaperone is involved in the biosynthesis of a molecule called zinc bacteriochlorophyll. And we think in those cases that it functions like a chelatase, like I showed you the picture of the iron chelatase and the magnesium chelatase, and we think this is actually a zinc chelatase. And so we have one project where we're looking at, can we take this bacterial protein and express it in plants and produce zinc chlorophyll? Because then if you can make a zinc chlorophyll, that is a way of enriching the edible tissues, at least of green plants. So here we're talking about not rice, but we'd be talking about spinach, for instance. It would have to be a plant where the edible tissue is the green tissue that you're eating. And that would be a case of where you could make something like that, where now the plant is very rich in zinc because it's, instead of making magnesium bound chlorophyll, it's making zinc chlorophyll. And so certainly I think those are within the realm of possibility. We just have to do it and see what happens first.
Cool. That's really neat. So for the transcriptics part of your work, what differences do you expect to see between pieces, sort of different parts of the plant, the root versus the leaves, for example?
Yeah, so with the roots versus the leaves, so for instance, the roots, you know, we kind of think of them as way stations of sorts. The roots are the place they take in the nutrients and they sort them and then they send them to the green tissues. And then the green tissues are the place where photosynthesis is happening. And so when it comes to a lot of these nutrient deficiencies, a lot of them are impacting photosynthesis negatively, which actually can lead to oxidative stress and all kinds of bad things. And so with a lot of these different conditions, we're expecting that in the green tissues, we're actually going to see a lot of how the plants are dealing with the stress of not having the nutrient. Whereas in the roots, we're seeing more of they're trying really hard to express the genes they need to import more metals. And so I guess it's mainly based more on like sink source relationships that we would expect to see different acclimation strategies in the different tissues.
Great, thanks. So let me see. Are there any plants out there that specifically require a lot more zinc than other micronutrients? And is that something you've encountered in the process of looking at zinc deficiencies?
Ooh, I bet there are. So I know there are these plants called zinc hypoaccumulators. And there are a lot of people who study these plants. No one knows necessarily why they do it, but there are plants out there that hyperaccumulate lots and lots and lots of zinc. Orders of magnitude more than any other plant. And it's thought that maybe those plants use this high levels of zinc to maybe ward off predation or someone, you know, eating the plant, or maybe bugs, you know, trying to eat the plant. So there are plants that seem to perhaps use that as like a defense mechanism. But surprisingly for us, so we're working with poplar and sorghum, which are two very different plants. I mean, one's a tree and one's a grass, but they accumulate in their green tissues the exact same amount of zinc and almost the exact same amount of iron. So across very diverse plants, at least, you know, not in these weird situations of hyperaccumulators, they pretty much stay within a window of these metals. But what we do see is that when we limit for one nutrient, so under iron limitation, for instance, the plants for some reason start to take up more zinc than they take up when they have tons of zinc. So there's some really interesting crosstalk between these nutrients that we're trying to understand.
That relates to the next question, which is related to both of the plants that you're studying, associating with mycorrhizal fungi. Hope I'm pronouncing that right, Steve. Which have been shown to uptake and translocate zinc and copper to plants. So do you think that these plants are going to be related to the expression of your chaperone proteins?
Oh,
like these fungi?
Yeah, so in the experiments we're doing, I am a reductionist at heart, and so I hate variables. And so like one variable is enough for me. And so in the experiments we're doing right now, we're doing like hydroponics with the plants. We're doing hydroponics, and we're saying, to the best of our ability, let's change one variable, which in this case would just be the concentration of the metals we're giving it. But of course, that's not real. That's very artificial. And so what we are moving towards is the next step is to do soil-based experiments. And the next step would then be to try and reproduce the microbiome of these plants that they would have in nature. And certainly to also have, constructed microbiomes that are, have fungi and other microorganisms that are known to be beneficial, especially when it comes to bioavailability of these nutrients in the soil. And again, I don't know. I would say that certainly the fungi would affect the bioavailability of the nutrients in the soil, but whether or not there is any signaling or communication between the fungi and the plants beyond that, I'm sure there is. And I don't know, but I think that's fascinating. And that's something we're working towards. We're just taking baby steps to get to that level of complexity.
Okay, thanks. So we have some questions that are about a bit more about the proteins themselves and some of the related proteins. So how greatly does the MAP2 gene affect cell growth among different species of plants? And like, can a cell or an organism survive off of MAP2 alone?
Okay, yes. So it's different in different organisms. Certainly. So in Saccharomyces that we worked with this experiment, excuse me, we were lucky because MAP1 is the dominant one, and the cells don't really care if MAP2 is there. And really, if you knock out MAP1, the cells do not grow. They do eventually. They're very, very slow. And if you knock out MAP1, MAP2, it's lethal. Now, if you move into humans, MAP2, for most cell types, MAP2 is dominant. And MAP1 doesn't necessarily seem to have, you can get rid of MAP1. It doesn't seem to matter so much, but MAP2 is the important one. And then that's the same for plants. So we've been doing work in Arabidopsis. Arabidopsis has, well, it's complicated, but Arabidopsis in the cytosol has 2 MAP2s and 1 MAP1. And so there's, if you get rid of MAP1, doesn't really care. If you get rid of one of the 2 MAP2s, doesn't really care. But if you get rid of both MAP2s, it's pretty much lethal. So there's definitely something we don't really understand about which type is going to be the dominant type. And maybe it has something to do with cofactor availability during evolution, and we just don't know. So what we've had to do in plants is we've actually had to knock out both MAP2s. The plants aren't so happy. And then we knock out the zinc chaperone, and then the cells really aren't happy. But yeah, we don't really understand why one type would be dominant over the other. And it could have something to do, like I said, with cofactor availability during evolution of zinc versus manganese.
So thinking like in terms sort of like the proteomic level, how widespread is zinc binding, zinc finger motifs in proteins and enzymes? And do you think that Zn1 or other zinc chaperones play a role in regulating availability and uptake of zinc across the proteome, more broadly speaking?
Yes, so that's perhaps the most surprising so far, is that there are hundreds of different types of zinc dependent proteins. I mean, eukaryotes especially are famous for, we have evolved an even bigger zinc proteome than prokaryotes have, because we've had lots of expansions in zinc finger proteins and in regulation and transcription. So there's tons of proteins that need zinc that are as essential or more essential than methionine aminopeptidase. So why does a methionine aminopeptidase need a zinc chaperone and no one else does? And so the answer to that is we think that perhaps it could be interacting with other proteins. We don't know what those other proteins are. We have done some experiments in yeast where we've done what's called proximity labeling, where we basically put a reporter on the chaperone and ask who is it coming in contact with. And so we have some ideas of some other proteins that it could be interacting with. In our proteomics work, where we look at the effect of the mutations, we see impacts on the ribosome that look like it's independent of its relationship with methionine aminopeptidase. And so the ribosome is the most abundant zinc-dependent protein in the cell. So yeah, I think it's more general, maybe, than just MAP1, but we'll see. So what's interesting with our stories, we did this work in yeast, and then I was contacted by a colleague who found the exact same protein MAP1, but in higher eukaryotes, in zebrafish and mice. So, we're not finding different client proteins. We're finding the same one, this MAP1. So I don't know, the jury's still out on that question.
So I think we have time for maybe one more follow-up before we move on to the career. Is that okay, Shelley? Okay, great. Along the same line, I'm gonna indulge myself and ask a question that I'm interested in, which is, you showed a structure of Zn1, right? Is that a structure of Zn1, or is it a homolog? And do we know, do you know how, like, what the zinc affinity and how many zinc binding sites and what the GTPase activity of Zn1 are? Because you kind of, it seemed like you were implying that a lot of that was inferred from knowledge of sequence rather than measurements of binding and function.
Yeah, so I didn't present everything we've done. Yeah, and we've done a lot, and we have a lot of questions, even based on what we've done. So yes, we've confirmed GTPase activity. It has GTPase activity. We see that GTPase activity is stimulated by the presence of divalent metal ions. We don't see specificity for zinc. So meaning you take the chaperone and you in vitro and you give it cobalt, manganese, nickel, zinc, it has GTPase activity. There's not really any preference. Here's the other thing we did that I didn't show you. In our metal transfer assay, we loaded the chaperone with zinc and then asked, can it metallate and therefore activate MAP1? It works. It's dependent on GTP. We did the same thing with cobalt. So we preloaded the chaperone with cobalt, and we see the same thing. So again, at least in vitro, when you're giving it a metal and preloading it a metal, there doesn't seem to be any specificity in the system. Now, we think that physiologically cobalt isn't a physiologically relevant metal. So we're pretty sure it's zinc, and the cobalt experiments were just for fun. But it opens questions, like we haven't definitively shown a reason why this chaperone would be specific to zinc. And that's not unique to us. That worries the entire metals field, that if you do any experiments in vitro, you just fundamentally don't see specificity or selectivity among the divalent, the first row D block. Okay, structure. So that was a homology model, but something I didn't talk about that was really cool is that the Baker lab came out with a paper end of last year where they used AlphaFold and Rosetta combined with coevolution studies to predict protein-protein interactions and then build complexes of proteins. And our chaperone and MAP1 came out of that study. So they were able to, completely independent of us, predict that these two proteins form a complex and then share with the world a structure of what that complex looks like.
That's nice.
It was amazing. And I looked at the complex and I'm like, this makes sense. It's a biologically meaningful complex. There are metal binding residues and different that we think is the metal binding site for the chaperone. And that site is perfectly positioned over the cavity that leads into the metal binding site of MAP1. It's just a beautiful structure. And it's really exciting. We've done, I didn't share, but we've done a lot of mutagenesis to look at the interaction between the two proteins and what disrupts interaction and all that. We have not done metal binding studies. And I haven't wanted to do those because I think it's a huge can of worms, because what we think is that this protein, since it's a GTPase, it probably is going to have different conformations and different metal binding affinities depending on its state. whether it's nucleotide free, nucleotide bound, GTP versus GDP versus who knows. So we just haven't done that yet, because I think that's going to take a lot of time to figure out on that kind of high resolution molecular scale, the actual mechanism of the metal transfer reaction.
Gotcha. Well, sounds like we'll have to stay tuned. Thanks so much for your answers to all of our questions and a great talk. We'd like to ask Dr. Blevy to spend a few minutes talking about her career path and kind of sharing that information with the students, and then we'll open the floor to questions about her career path and opportunities at national labs and things of that nature.
Right. I just put together some simple slides. Let me share my screen again. For my career path. So my career path started out in Florida, and then I went to Los Angeles, and then I moved to New York. Sunshine, sunshine, and then snow. So I started my career, I grew up in Florida. And I actually grew up on a farm back in the 80s. We grew tobacco, we grew chickens, and I wanted to share my statistics. So everyone is born a statistic, and statistics don't matter. But I think it's interesting to think about what is your statistic, because it provides you context with what you have overcome to get to where you are. So for me, I was born to a teen mother. And the statistic, is that children of teen mothers are more likely to repeat grades. They're not likely to complete high school. I was raised on a farm in rural America, less likely to even get a bachelor's degree. And then also I was, I'm a girl, and I was, girls, at least back in the 80s, were much less likely to, complete college. But, statistics don't matter, of course, especially not in this country. And so I got a Bright Future Scholarship to go to the University of Florida, which is a great scholarship program that the state of Florida has, that basically says, if you get good enough grades, then you can go to college for free. And it was when I was an undergraduate that I did a research project, and I encourage anyone who's interested in science, which if you're sitting in this seminar, you must be interested in science, and you should take the opportunity to do an undergraduate research project. And for me, it just really solidified my passion for science and learning that I can be curious about the world around me and do something about it, which is research and experimentation. And I actually didn't know what I wanted to do, but I ended up, I guess, kind of fell into it because my undergraduate advisor was like, what do you want to do? And I'm like, I don't know. And he's like, well, why don't you go to grad school? You can come to grad school here at UF. And I'm like, okay, sure. And so I kind of fell into it and went to graduate school at the University of Florida, and just absolutely loved it. I had a great mentor. Something that I certainly didn't know, and I always like to share with people, is that if you're thinking about going to graduate school, remember that it's not, if you pick the right school, it's not really going to cost you anything. You're going to get paid a stipend, and your tuition is going to be waived at most departments. So there's really no financial reason not to go to graduate school. And I loved graduate school, and I was there for about 4.5 years and published 8 publications. I then was lucky enough to get a postdoc position at UCLA. So I traveled all the way across the country, as far away from Florida as I could get. And I just, again, once you find what you love to do, I mean, that's what's most important. And if you love what you're doing, you're going to be successful. And so as a postdoc, I was there again about 4.5 years, published 16 publications. I was actually lucky enough to receive an NIH fellowship. And then I love this picture here at the bottom. This is me. I received a postdoc research award from Paul Boyer. He's no longer with us, but he was a Nobel laureate who got his Nobel Prize in a lot of the fundamental photosynthesis research. And I actually received an award that was in his honor. And so that was very cool. And then I made it, as a postdoc, I just loved the idea of having big teams of people working on big experiments with that are really important for society. And so an opportunity came up to build a program from scratch at Brookhaven National Lab, and I took it. And I've been there about 7 years now and have been able to build a team of researchers that were working together and doing fun stuff. And that is my career path.
Wonderful, thanks for that. We've got a bunch of questions rolling in. So the first one is, were you a first generation college student?
So I technically no, because my grandmother, when I was about 12, went back to school. And so she, as a grandmother, went to university and got a degree. So I am perhaps maybe not a traditional first generation, but I'm not a first generation.
How did you decide? But so it sounds like kind of.
Kind of, yeah. Like,
yeah. So how did you decide you wanted to go to college? Like, how did that path evolve from growing up?
I don't know. I mean, growing up, I never thought about not going to college. And maybe that's just because the people around me, my mother, because I was always really good at school, there was just the assumption that that would be the path I would take, that of course it would be a waste if I didn't go to college. And so I've actually never thought about it, because I think I just always knew I was going to go to college. Yeah.
Well, so you grew up on a farm, and one of our participants comments that they know a few people that grew up on farms and heavily pursued careers in science. Do you think that early influence of living on a farm seeded some fascination with life processes and influenced your career path?
I wonder, maybe, because certainly, like raising your own animals and feeding it, like that whole process, perhaps. I mean, I know, at least for me, we were in a county that the University of Florida has a really great extension program where they have extension officers and agents who work for the university, but then they have offices out in all the rural parts of Florida, where the farmers are interacting with these university agents. And so from a very early age, I had that connection to the university, and I had that connection to these professionals who had pursued scientific careers and then took that knowledge and understanding back to the farm. And so I think that influenced me as well. To have these interactions with these extension agents and to understand the process of how science can help farmers and agriculture. And certainly maybe not consciously, but now that I'm thinking about it, perhaps that's always been a part of my experience growing up, and perhaps influenced the trajectory I have now taken.
So what would you say is the biggest challenge you faced to reach where you are now? And do you have any advice for undergraduate students that might want to pursue research?
Yes, it's not easy. So, especially graduate school is not easy. And we always joke that when it comes to getting a PhD, it's not about how smart you are, it's about your endurance. And if you can just work hard and make it through. So I think there's a misconception that graduate school is easy, and it's an easy path, and it's not. And it takes a lot of hard work. And so I think it's important, if you decide to go to graduate school, having that mindset, because a lot of people will leave after a year or two. I had a friend in graduate school who she really wanted to go to medical school, and she got into medical school, but her parents said, oh no, medical school is going to be too stressful and too much work, so you should go to graduate school. And so, we would be crying over experiments, stressed out and laughing because, maybe medical school would have been the easier route sometimes. But I think, but again, I come back to this passion. If you love it, and you enjoy what you're doing, then it doesn't seem like so much work.
So that leads directly into one of the other, I guess, a couple of the questions, which I'll sort of put together for the sake of time, which is like, you're clearly passionate about your work. how did you decide to work with zinc? Were there other topics that interested you? Like, how do you end up narrowing down on this one specific thing? And do you have like some dream experiment or other system that you always wanted to like try?
Yeah, I mean, so I presented about zinc, but I should say I have other projects and other publications that we're working on with iron and heme and copper, and lipid biosynthesis, and some synthetic biology projects in algae. And I presented on zinc, but I have way too many things that I'm doing because I'm just generally very curious about what's unknown about biology. And especially we have access to genomes. And so genomes are these wonderful maps that tell you how much you don't know about biology. And I just find that exciting and fascinating. And I love coming upon a gene or protein family that we don't know how they're functioning or what they're doing in the cell. And so, trying to figure that out is just what's really exciting. But I mean, I guess, I could have technically worked on anything, and the influences come from graduate school. So when I was a graduate student, I had an option of different projects to work on. I actually was in a group that worked on tRNA modifications, which are these modifications that are needed to help tRNA molecules fold properly. And I could have worked on that, but my mentor presented me with this project, and she was like, I have some preliminary data. I think it has something to do with zinc. Do you want to, work on it and see what happens? And then I just really enjoyed it. And then as a postdoc, I didn't work on zinc at all as a postdoc, actually. At UCLA, I worked on copper and worked on copper chaperones, and made some discoveries there that were just really fun and exciting and rewarding. Because I think so passion is part of it, but the other part of it is when you discover something and you realize that you are the only person who knows it, that no one else in the world knows it, you just figured it out. That is like the best feeling in the world. And then when you get to the point where you then get to share that discovery with everyone else through publications, through seminars, it's just, I don't know, it's intoxicating and addictive to get to that point. But yeah, I mean, research is definitely at the point where you can reinvent yourself and your research can go in new and different directions. And so even though I'm working on zinc and iron now, who knows what I might be working on in 10 years.
I think the last question we have time for is about the SULI program. Can you give us a little bit more information about how Rowan undergraduate students might get involved? Is there a possibility of doing research in your group through that? How long is the commitment? Like, what is looked for in applicants? Can you give us some insight there?
So I can give you from my own experience. So what I find for me is that if you are interested in doing a project, contact a researcher directly. Everyone's emails are online, and you can contact the researcher directly and say that you're interested in doing a research project, and read about that researcher's research beforehand and make sure that that's the type of researcher and the type of research you're interested in. And then you can introduce yourself and say that you're interested in a project. And that's how all my interns end up coming through that route, where potential interns will contact me. And then from their email, I'll get a sense as to whether they're actually interested in the research I'm doing or they're just looking for anything they can get. Because like I said, passion and interest in it is a big part of research. And so you want to be doing an internship and a research project that you find interesting and you find stimulating. And so I recognize that as well. And I want interns that aren't just doing it as something to put on their CV, that they actually are interested in the project. Yeah, and there's an office at Brookhaven called OEP, and I put the website and I can send it around, and there are professional people at Brookhaven who have all of the details about the SULI program.
Do you look for those students to have obviously interest, and genuine interest sounds like key for what you look like for in those interests. Do you also look for any specific skills?
Not usually. I mean, usually what I try to do is I try to get an understanding of what unique skills an intern might have, and then tailor a project to take advantage of those skills. So for instance, I have computational projects, bioinformatic projects, experimental projects. You know, if an intern comes to me and says, you know, they love Python and they want to do a bioinformatic project, then I'll say, yay, here's a project we'll do. Or if I have a student that comes to me and says, oh, I really love structural biology, I want to learn to crystallize proteins, then I'll be like, yay, you can do that too. So I try to be pretty flexible with the actual projects, and I try to talk with interns to see what they know how to do, what they would like to know how to do, and what they find interesting. And I try to tailor projects to the intern.
That's very cool. Thank you again so much for sharing with us about your research and your career path. We really appreciate your time. Thank you everyone for joining us today. I hope everybody has a great weekend. And thanks to Dr. Thomas for sort of sharing the opportunity to talk with you with the MCB department. Really appreciate it.
Thank you guys. I appreciate it. It's been fun.
Okay, we have stopped recording. Thank you so much. That was amazing.
That was awesome.
Yeah.
Cool. Yeah. Thank you for the invite.
Yeah.
It was great.
I was actually kind of scared that I wouldn't understand what you were doing, but you did such a good job of explaining everything in ways that undergrads could understand. So I really appreciate your approach to.
Okay, good. Thank you. Yeah, because I mean, other than interns, we don't have undergrads or graduate students. So a lot of times I worry that, you know, you get stuck in this like different language when you're around people and you're always talking about the same things, and especially with the Department of Energy, we have acronyms for everything. And so so thank you. I really appreciate you saying that. That I haven't completely lost touch with.
Yeah, the level was not only was the level really spot on for undergraduates, the audience here bridges everybody from really basic biologists, people who are interested in ecological questions, all the way to structural biologists. Like my entire lab was in there, and we're a protein biophysics lab. So I mean, there was a little bite for everyone, and it really impressive.
Oh, great, thank you.
Yeah, and I was really glad that you shared the reach out to people directly for SULI. Yeah. I didn't even know that was a thing for SULI positions, and I imagine it varies pretty heavily from you know, sort of, and from the different sites. But we've had a couple students from the physics department get positions just by doing sort of the cold application to SULI. One of them got a position at